Regenerative medicine is one of the fastest growing fields with new papers published every single day. As a busy mom of two, I am guilty of not making the time to keep up with the research, and I think a lot of other veterinary professionals are in the same situation. My name is Dr. Adrienne Wright, and welcome to Sit, Stay, Learn, a short podcast where I review regenerative medicine uses in veterinary science. Each episode, I break down a paper. We spend 10 to 20 minutes here just giving you the details you need to know to feel like you're staying up with the research. This paper was sent to my team out of excitement for our own Canine ACV product that you all knew and loved that we had to stop offering earlier this year. But on a good note, this paper felt very fitting to review on the tail of a big accomplishment. We finished enrollment earlier this month ahead of schedule for our clinical trial comparing the treatment of osteosarcoma using our Canine ACV plus checkpoint inhibitor against carboplatin, and we're ahead of schedule. We are so excited at the progress of the trial, and we'll share it with you as soon as we can. But this review paper is titled Autologous Cancer Vaccines, a Precision Immunotherapy Strategy for Veterinary Cancer Patients. And it was published in Anti-Cancer Research in August 2025. I think this group did a really nice job of laying the land for emerging treatments of veterinary cancer immunology, veterinary cancer immunotherapy, and explaining the differences in those therapies. So, let's dig into this paper a little bit. Personalized medicine is a strategy that provides a specific patient a specific course of treatment based on a variety of factors that are unique to that patient. These factors can be clinical or genomic, genetic, environmental, etc., etc. And this approach is really logical. Personalized treatment will provide better outcomes compared to a one-size-fits-all approach, which is historically how care has been handled. And the pharmacogenetic approach for cancer stems from the success with Gleevec. It was a selective ABL inhibitor that improved 5-year survival for people with chronic myeloid leukemia from 30% to 96%. Gleevec started with one groundbreaking find by doctors Nowell and Hungerford in 1960 when they examined cancer cells from 2 patients with chronic myeloid leukemia and noticed that one of the 46 chromosomes was abnormally short. They found more patients with the shortened chromosome, and this chromosome was later named the Philadelphia chromosome, and 95% of patients with chronic myeloid leukemia have it. Over a decade later, they discovered that the Philadelphia chromosome forms when 2 chromosomes break and then the pieces swap places. And when it forms, 2 genes that are normally separated fuse, the BCR-ABL fusion gene. And when this gene forms in the blood cells, you get chronic myeloid leukemia. And this is the basis of Gleevec. It was approved by the FDA in 2001. So, what came from Gleevec's success was an idea that you can match a targeted inhibitor to an identified mutation, and then that's going to give you the same success. And you rinse and repeat for every cancer. But obviously, cancer is not that easy. The generalization did not take into account that there are multiple actionable targets that often drive cancer growth and metastasis. So, obviously, the Gleevec approach doesn't apply to all cancers. And this lesson is what actually renewed interest in cancer vaccines, because a vaccine, more specifically an autologous vaccine, is an ideal solution to multiple targets because it incorporates multiple antigens that are relevant to that patient's own tumor. So, what are tumor antigens? Well, it's a somatic mutation of tumor cells. That's what's responsible for the extreme antigenic heterogeneity between tumors, even of the same cancer. The antigens have 2 main categories. You have tumor-associated antigens and then tumor-specific antigens. The associated antigens are normal self-proteins, and because of immune tolerance, these are not sufficient to create an effective immune response. So this would be like HER2 new. The tumor-specific antigens are the mutated novel proteins that have no central immune tolerance and are highly immunogenic. They include neoantigens, and they are great targets for immunotherapy because of their cancer specificity and immune recognition. There is a third source of antigens you don't hear as much about, and those would be from the tumor stroma. This is a mix of cells and extracellular matrix that support cancer growth and metastasis. But even more interesting to me is that the tumor stroma antigens also contribute to local immunosuppression, and they restrict T cells from getting into the tumor. So the authors make the claim that I think all of us are thinking at this point. If you can get an immune response directed against the cancer cells and the stroma that is doing the immune suppression, then that would be the best therapeutic strategy. It should also be noted that tumor stromal cells are much more stable, both antigenically and genetically, than tumor cells because they're not going through that same somatic mutation. It makes them extremely valuable in a vaccine. And so any vaccine that's going to include the whole tumor, like ours, would have those tumor stromal cells. So, overview of cancer vaccine. In general terms, the goal of the cancer vaccine is just to expand the population of effector T cells that are capable of destroying that tumor. There are a few vaccines that have already been studied in dogs. I'll share a very quick one, but they also discuss another one no longer in commercial development if you want to check out the full paper. So, Oncept is made with tumor-associated antigens. It has a DNA plasmid for tyrosinase, and it helps overcome a dog's central immune tolerance to tyrosine, which is associated with melanoma. It was reported to be well tolerated. This is a xenogeneic vaccine, meaning from another species to a dog. There are some conflicting reports on this product out there, and I'm going to be honest, the benefit of this one's a little murky. But also, since it is using tumor-associated antigens and tyrosine is only associated with melanoma, then this vaccine is limited to only the one cancer. But it has been reported being used for melanoma in species other than dogs. So, this is what makes the tumor-specific antigens a better choice for vaccines, specifically neoantigens. So neoantigen-based vaccines may increase therapeutic efficacy and lower the probability of an autoimmune adverse event when compared to vaccines using just the tumor-associated antigens. Vaccines tested in murine models have shown tumor reduction and increased survival with low toxicity. In humans, they've also been really well studied. So there's over 100 clinical trials, and they've demonstrated CD8 T cell expansion as well as increased survival. But determining neoantigens is tough, and it requires sequencing of the tumor genome to identify express protein coding sequences against a normal background, and then even more testing. So prediction of meaningful neoantigens has been weak. There are a couple strategies that have been described in veterinary medicine, one for melanoma with binding to a unique DLA, the other uses frameshift peptide arrays. And I think these are interesting approaches, but this is a very resource-heavy undertaking with difficulty predicting, and it may be better in veterinary medicine to maybe attempt a cancer type versus an individual vaccine with these strategies for each patient. So, based on the fact that efficacy is limited for vaccines using tumor-associated antigens, and challenges in predicting neoantigens for veterinary, the clear argument to be made is autologous cancer vaccines, or ACVs, are the way to go, which is exactly what ARNet has in an ongoing clinical trial. The groundwork for cancer vaccines is quite broad. It dates back to the 1930s. This is not a new concept. But briefly, an ACV is an autologous vaccine derived from that patient's tumor, which then exposes the immune system to tumor-associated antigens, as well as tumor-specific antigens, neoantigens, and possibly stromal antigens. These vaccines are highly specific. They present the immune system with the exact antigens of the tumor. They are also, in some cases, not bound to a species or a tumor type, which is really what makes them so valuable in veterinary medicine. ACVs are usually inactivated whole or lysed tumor cells or tissue, and then some formulations have tumor matrix, associated antigens, but this is all going to depend on the production protocol. Common methods are enzymatic digestion of the whole tumor and then inactivation of the cells. Usually you use UV irradiation here. Some do repeated freeze-thaw, some do mechanical digestion. It varies. Some methods do cell culture. Ours does not. I think that the heterogeneous pool of cells and antigens is what makes it so effective rather than a culture-adapted cell line, but also those have their place. But they do lose some of that heterogeneity, and I think that's what makes it so special. Improving the immune response with these vaccines is critical, so most methods use an adjuvant. Early work used bacterial adjuvants. Freund's complete adjuvant has also been used in dogs, which is a very popular nonspecific adjuvant with mycobacteria. It's been around for decades. There are other vaccines like Ardent's that uses unmethylated CpG motifs. CpGs are short synthetic oligodeoxynucleotides, or ODNs, that are recognized by a toll-like receptor 9, and they induce a Th1-dominated immune response. The CpG ODN that we use is canine specific, so it even further enhances that immune response. There are other methods to improve immune response. Another one is a plasmid transfection of cancer cells to induce expression of human granulocyte monocyte colony stimulating factor, IL-2 or M55. These transfected vaccines have been administered to horses and dogs, but the majority of the work is done either in a small number of animals or they lacked an objective immune response. So there are some exceptions, but this doesn't seem to be like the commercial approach right now. Another option explored was dendritic cell vaccines, which involves harvesting dendritic cells from the patients using leukapheresis, exposing them to tumor cell lysate, then you infuse them back into the patient. They've been studied in several cancers for dogs and horses. There's a similar approach with a very similar workflow using CD40 activated B cells loaded with tumor RNA in dogs with lymphoma. Overall, clinical data is limited, but they did demonstrate an immune response. But again, adding the leukapheresis component makes this technology unattainable for many pet owners because access to this equipment is really limited. An interesting study in canine glioma showed that autologous tumor mRNA packaged into lipid particle aggregates improved median survival time to 139 days compared to 35 days for their historical controls. Although this was a very small sample size, those results are promising. But as the authors point out, there are over 50 years of data using ACV in veterinary medicine. And although many of the studies are small and retrospective studies and inconsistent in reporting adverse events and clinical outcomes, blah, 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 if you take all the studies together, it's convincing that ACVs do have a good safety profile and potential efficacy. They cover many of these studies in Table 1. We don't have time to cover them all, but one that I want to highlight is Ardent's pilot study that was published back in 2016. That was shortly after we started commercially offering our vaccine. This pilot study was done on multiple cancers, and we did demonstrate a detectable humoral response and increased survival time, which is something that we continued to see over the next 9 years until we stopped the commercial service. This vaccine, with some improvements and additions, is the vaccine being studied in the clinical trial that I mentioned earlier. And I do want to note here that we were required by the USDA to stop the sale of our experimental product back at the end of June 2025. So with this clinical trial, this new and improved vaccine may be able to return to the market, and that's what we're all excited about. But there are other studies highlighted, and they go on just to support the fact that immunotherapies are safe and promising for the treatment of cancer in veterinary medicine. Another interesting point that they bring up is that ACV may be useful in the adjuvant setting for patients with established mets. A recent retrospective study described the use of ACV in dogs with metastatic hemangiosarcoma following a splenectomy. The study demonstrated that dogs treated with a weekly dose of mechanically dissociated tumor tissue, inactivated and combined with cysts for a total of 3 injections, had a significantly longer median survival time of 142 days compared to surgery only at 41 days. It was a huge increase. But this survival time is similar to that of dogs treated with the max tolerated dose of chemo. And the treatment group was only 8 dogs. But data is still promising, and there were no adverse effects noted, which I think you'll see is a recurring theme here. Obviously, you can't discuss cancer immunotherapy without talking about Elias gaining full approval in March of 2025 for their ECI product. It was based on their pivotal efficacy and safety study for dogs with osteosarcoma. ECI activates cancer antigen-specific T cells, which are harvested later by leukapheresis, expanded, and then infused with recombinant IL-2. In their single-arm study, median overall survival for dogs was 415 days, compared to historical median survival time of 134 days for amputation alone. They have updated this data to show combined use of ECI with chemo. I think we'll probably cover that in a future episode. I think ECI getting approval was a huge win for the industry, but there is the limitation of treatment centers due to that leukapheresis step. Even some oncology practices are not able to treat, or some of the oncology practices are able to treat, but they're having to send their patients to a secondary location, which may not be convenient for the owner, sometimes not even feasible. So to me, this is what excites me most about our vaccine potentially coming back on the market, because any general practitioner can participate in the service. But like I mentioned, Elias did publish data on their ECI plus chemo, and that combo of ACV plus chemo is not really a novel approach, but that's what they most often use with lymphoma due to how fatal it can be. They cover data from several studies with lymphoma, all with promising data and promising survival times compared to the controls. But the last section of immunotherapy they cover, which I'm probably most excited about, is the addition of checkpoint inhibitors. So ACVs are used to stimulate an effector T cell response, while checkpoint inhibitors are used to overcome local tumor-driven immunosuppression, which then allows the CD8 positive T cells to interact effectively within the tumor and kill the cancer cells. This strategy was demonstrated by Dr. Michael Olen's group at the University of Minnesota in a pilot study of dogs with high-grade glioma. These dogs were treated with tumor lysate and then checkpoint inhibitor, or checkpoint peptide rather, CD200 ARL. The overall median survival time of dogs treated with vaccine plus CD200 was 12.9 months, compared to 6.8 months for vaccine treatment alone. Historical data of surgery alone has a really dismal median survival time of 66 days, and in the control group, no dog survived longer than 7 months. Another interesting note is that the tumor progression was the cause of death of 60% of dogs treated with vaccine and CD200, compared to 87% of dogs in the control group. So, to recap the study, adding the checkpoint peptide CD200 to the vaccine almost doubled the median survival time compared to dogs treated with vaccine alone. But why is this exciting? Because Ardent has licensed the CD200 checkpoint peptide, and it is being tested in combination with our vaccine that you all knew and loved for our clinical trial. And of course, there are other CPIs on the market. Gilvetmab was a monoclonal antibody, or is a monoclonal antibody against PD-1, and it was the first veterinary CPI and received conditional licensure. But of course, with any technology, there are challenges, and Gilvetmab is no different. It is quite expensive, and it's hard to get with a limited supply, but I think that will probably change over the next few years. The last thing I want to note is the authors did a fabulous job of discussing the challenges with cancer immunotherapies. So they've presented all this data and they set it up so you're like, yeah, immunotherapy is the answer to tackle cancer, and then they hit you with the challenges. So as with any treatment, there is no 100% success rate. So they talked about successful studies, but then they're the ones where they failed to produce a clear clinical benefit. And the ability to predict who's going to benefit, I think, is one of the biggest hurdles to a study. But there are so many factors that make this difficult. So several we covered as we discussed the intricacies of this technology, but you have the preparation of the vaccine itself. You have the adjuvant choice, and putting that out there because I think it is important. You have dosing, and that includes the amount, the route, the frequency, tumor characteristics, patient characteristics, and the list goes on and on. And this is even more complicated because the canine immune system is understood even less than humans. And there are no validated tools, which we discussed pretty heavily in how you measure pain for clinical trials in our previous episode. So there are no validated tools to study an immune response in these animals. But despite the challenges, ACV truly represents one of the most attractive cancer strategies out there. And this is the recap of why. Because they use cancer material from that patient, and they likely have the most immunologically relevant antigens to be presented to the immune system. They seem to be well tolerated, and that's independent of cancer type, of formulation, and species. But they are not readily available like an off-the-shelf therapeutic. They take time, they take labor. There is no economies of scale here, and because of that fact, the innovation really is being led by small animal health companies like us. And I think that's pretty amazing. Can't wait to see the innovations that come in the next 5 to 10 years. But overall, I think this was a really insightful article. It left me with a lot to think about. What are your thoughts? I would love to hear them. And please keep requesting copies of these papers and sending me papers to review. I want to help you feel like you know and understand the science that is being put out there. For the text version of this episode, you can head to our blog. And if you have questions on this episode or a paper you'd like for me to review, you can email me at adrienne@ardentanimalhealth.com. I hope you all have a wonderful week.